The last universal common ancestor of everything alive today was likely a hydrogenotrophic acetogen, which lived between 3.7 and 3.5 billion years ago. It's from this ancient metabolic process that all dissimilatory metabolic systems alive today are derived. But how did these come about from such a seemingly humble beginning? Well, for one, with the advent of the first ATP synthase enzymes, life could replenish phosphodiester bonds as its energy currency. From here, early forms of heterotrophy such as fermentation could have come about. Prior to LUCA, organisms could use exogenous reactive phosphorus species such as pyrophosphate to power the synthesis of biomonomers like sugars from simple organic molecules. These pathways could then be reversed, breaking biomolecules down into simple organic molecules to power the replenishment of the phosphodiester bonds in ATP. This would have given rise to the first form of substrate-level phosphorylation. As discussed in the What Was the First Organism Like and From the First Organism to the Last Common Ancestor videos, abiotic analogs of glycolysis in the pentose phosphate pathway have been identified. The abiotic pentose phosphate pathway runs reductively but not oxidatively, indicating that it was originally used for biosynthesis and later reversed to drive energy metabolism. Furthermore, many of the enzymes that are responsible for catabolic fermentative processes such as glycolysis also catalyze anabolic processes such as gluconeogenesis, strongly suggesting that the two pathways share a common ancestor. In order to regenerate the all-important coenzyme A, the phosphotransacetylase and acetate kinase enzymes used at the tail end of the Wood-Young-Dahl pathway could be used, which would produce acetate as the end product. This would have the added benefit of producing more ATP, but there is still the question of the NADH. Substrate-level phosphorylation will consume NAD, and it could be replenished by using to synthesize other biomolecules, but if the organism was relying on fermentation to power its energy metabolism, it can only go so far with this. Eventually, it will run out of these oxidized electron carriers, meaning the first fermenters could only use it for a limited time. To regenerate NAD, the acetyl-CoA could be reduced to ethanol, or the pyruvate could be reduced to lactic acid instead of being reacted with coenzyme A to form acetyl-CoA. While these couldn't produce as much ATP per sugar metabolized as acetogenic fermentation, thanks to NAD regeneration, they could continue indefinitely so long as the organism was provided with glucose. Although, one way early fermenters could have their cake and eat it too was to use a hydrogenase enzyme to oxidize the NADH, producing hydrogen as a fermentation product. Going back to the autotrophic processes, the other dissimilatory metabolism that uses the Wood-Young-Dahl pathway is methanogenesis. The overall sequence of reactions in the early steps of methanogenesis and acetogenesis are quite similar, but with significant variation in the chemicals used. Both start with a reduction of carbon dioxide to formate, but instead of priming it by binding it to a phosphate group, methanogens do so by binding it to a methanofuran. And instead of transferring it to a tetrahydrofolate, methanogens bind it to a related molecule known as tetrahydromethanoterin, or H4-MPT. This molecule is similar to the tetrahydrofolate employed by acetogens in that they both possess a pterin functional group. In biochemistry, this molecule type likely originally came from folate or vitamin B9. There's also a series of steps where the carbon is reduced to a methylene and finally a methyl group. This is facilitated by hydrogenase enzymes, which take electrons from hydrogen molecules, and cytoplasmic electron carriers, which deliver them to the THF and H4-MPT for carbon reduction in acetogens and methanogens. However, instead of using NADH, methanogens use another electron carrier exclusive to prokaryotes known as F420. While functionally similar to NADH, which is a niacin derivative, they structurally resemble flavins or vitamin B2. Once the carbon has been reduced to a methyl group, it can be reacted with carbon monoxide, which was produced by reducing a carbon dioxide, and coenzyme A in the carbon monoxide dehydrogenase acetyl-CoA synthase enzyme complex, producing acetyl-CoA. This will then be fed into biosynthesis pathways to be assimilated. If it's used for energy metabolism, it will continue to be reduced. This is done by reacting the methyl H4-MPT with coenzyme M, producing methyl-CoM and releasing the H4-MPT. The methyl group is bound to coenzyme M by a thioether, and when the methyl CoM reacts with coenzyme B, it forms a disulfide bond with it, releasing the methyl group as methane. Coenzymes M and B are regenerated by the heterodisulfide reductase enzyme, which uses electrons to reduce the disulfide group to a pair of thiols. The electrons which do this again come from a hydrogenase, but are flown through another ion-pumping complex, along with the RNF complex, which uses electrons which reduce the formula H4-MPT. From the ion-pumping complex, the electrons are shuttled to the heterodisulfide reductase enzyme by ferredoxins, but are shuttled from the hydrogenase to the ion-pumping complex by electron carriers within the membrane. This feature is not observed in acetogens, and one variety of carriers exclusive to methanogens is methanophenazine. Like F420, this has a redox-sensitive functional group similar to flavins, suggesting these coenzymes were derived from vitamin B2. To facilitate the flow of electrons through membrane-bound enzyme complexes, methanogens possess serohemes, porphyrin-based molecules with an iron in the center. These were likely originally modified versions of cyanocobalamin or vitamin B12, with the cobalt ion switched out. These are critical for the diversity of dissimilatory metabolisms, as we'll soon see. In the From the First Organism to Luca video, we discussed how hydrogenotrophic acetogenesis could be seen as a convergence of carbon assimilation and energy metabolism. If the replenishment of ATP by the production of acetate was the original uncoupling of these two processes, the downstream part of the methyl branch of the Wood-Young-Dahl pathway used by methanogens can be seen as a further decoupling. In that sense, evolutionary innovations like membrane-soluble electron carriers and serohemes would be building on the ability to harvest environmental redox chemistry to drive ATP replenishment. Experiments which have knocked out the genes responsible for the downstream part of the methanogenesis pathway have shown that the organisms will instead produce acetate from acetyl-CoA, further indicating that methanogenesis evolved from acetogenesis. Apart from autotrophic methanogenesis, there are two heterotrophic versions of this pathway as well. The first is acetoclastic methanogenesis, which splits an acetic acid molecule into methane and carbon dioxide, and it does this by inputting the acetate at the end of the Wood-Young-Dahl pathway and running it backwards up the carbonyl branch while going forward on the downstream part of the methyl branch. The other variety is known as methylotrophic methanogenesis, and this oxidizes a carboxyl group on one organic molecule to carbon dioxide and uses the electrons to reduce a methyl group on another organic molecule to methane. It does this by running the methylated compound forward on the downstream part of the methyl branch, while running the carboxylated compound in reverse on the upper part of the methyl branch. To be more specific, acetoclastic methanogens use the acetate kinase and phosphotransacetylase enzymes to produce acetyl-CoA from acetate, coenzyme A, and ATP. The carbon monoxide dehydrogenase acetyl-CoA synthase enzyme then reacts the acetyl-CoA with H4-MPT, forming methyl H4-MPT and carbon monoxide while releasing the coenzyme A. The methyl H4-MPT is then reacted with coenzyme M, which is then bound to coenzyme B, releasing the methyl group as in other forms of methanogenesis. The heterodisulfide reductase enzyme again regenerates coenzyme M and B, and the electrons to do this come from the oxidation of carbon monoxide to carbon dioxide. Meanwhile, methylotrophic methanogens react the methyl group with coenzyme M and the pathway proceeds from there. The electrons used by the heterodisulfide reductase enzyme come from the upstream part of the pathway running in reverse. Apart from using carboxylated compounds, other groups in various redox states can be input in the upstream part of the methyl branch and be incrementally oxidized to carbon dioxide by the reduction of multiple methyl groups to methane. So, from acetogenesis to hydrogenotrophic methanogenesis, and then acetoclastic and methylotrophic methanogenesis, we can see how new pathways can come about by adding on to parts of existing pathways or altering the direction of other portions. Early methanogens likely lived in volcanic vents, and here sulfur dioxide from the magma will react with water to form sulfite. This is highly reactive and thus is very toxic to cellular life. It was recently discovered that some methanogens have enzymes which resemble the dissimilatory sulfite reductase used by sulfur-respiring organisms. These are called DSR-like proteins, or DSRLP, and these have a single subunit, while the DSR used by sulfur reducers have two in their quaternary structure. Two extant groups of DSR-like proteins have been identified, and The type 3 has 2 iron-sulfur clusters, while the type 1 doesn't have any. Meanwhile, the DSR used by sulfur-reducing microorganisms has only 1 iron-sulfur cluster per subunit. DSR LPs detoxify sulfites by using an as of yet unknown cytoplasmic electron carrier to reduce them to hydrogen sulfide. The original version of this enzyme wouldn't have had any iron-sulfur clusters. At some point, the B iron-sulfur cluster was added, which would have more readily facilitated the flow of electrons to the sulfite. One lineage had a second iron-sulfur cluster added, which ultimately gave rise to the type 3 DSR LPs. Meanwhile, another lineage underwent a gene duplication, and these would become the A and B subunits of the DSR enzyme. The DSRB subunit lost the second and third arginine-lysine binding sites. The DSRC subunit, which connects the active site to the electron transport chain, is homologous, i.e. shares a common ancestor, with the E subunit, which performs the same function in the aforementioned heterodisulfide reductase enzyme. This suggests that the DSR proteins originally came about when a DSR LP became hooked up to the energy-producing electron transport chain via the HDR, and sulfite, instead of carbon, became the terminal electron acceptor for the oxidation of hydrogen. This would have given rise to the first form of dissimilatory sulfur metabolism, and would give early sulfur reducers an edge, because sulfur is more electronegative than carbon, and they can thus get more energy by using it to oxidize hydrogen. We see something vaguely similar to this in a variety of type 3 DSR-like proteins known as FSR, who can detoxify sulfite at a greater rate since it is coupled to an F420 dehydrogenase enzyme, and thus gets its electrons from one of the most abundant carriers in the cytoplasm. This can be seen as a window into the past to look at how dissimilatory sulfur metabolism first evolved billions of years ago. If we look at the phylogeny of the DSR proteins, it largely aligns with the 16S rDNA phylogeny between bacteria and archaea, but with some of the DSRs belonging to early diverging thermophilic bacteria branching off first. This suggests that the DSR enzyme first evolved shortly after LUCA in a hydrothermal setting. And since sulfur is more electronegative than carbon, the sulfite could be used as an electron acceptor to oxidize organic molecules, which would in turn give birth to the first form of respiration. How exactly did it come into being? Well, we can never know for sure, but many biosynthesis pathways involving using one biomolecule to reduce another. Instead of reducing the other biomolecule, the electron carrier could instead shuttle it from the oxidized one to the electron transport chain, with sulfite as the final electron acceptor. This would explain why sulfur aspirers today don't have mechanisms to completely oxidize organic carbon, and instead have a hodgepodge of enzymes which partially oxidize various organic molecules. Although being able to oxidize organic carbon would give the first sulfur reducers access to more energy to drive ATP replenishment, the utilization of sulfite would be limiting due to its toxicity. Early microbes could circumvent this problem by using other less toxic oxidized sulfur compounds. In hydrothermal settings, one of these would have been thiosulfate. The enzyme which reduces it to sulfite and sulfite is in the molybdotierin oxidoreductase enzyme family. LUCA seemed to have one of these, known as arsenite oxidase. The respiratory arsenate reductase enzyme is also in this family, but surprisingly it's more closely related to the thiosulfate reductase enzyme than it is to the arsenite oxidase. Furthermore, thiosulfate reductase can also reduce arsenate to arsenite. One possible origin for the thiosulfate reductase enzyme is an arsenite oxidase enzyme which became bound to the main electron transport chain, where the influx of electrons caused it to operate reductively. Because of the similarities between arsenate and thiosulfate, it could have instead reduced thiosulfate, becoming the first thiosulfate reductase enzyme. This proto-thiosulfate reductase would then give rise to not only the modern thiosulfate reductase, but also the respiratory polysulfide, tetrathionate, and arsenate reductase enzymes. Of these, polysulfide reductases are the most closely related to thiosulfate reductase, and are often lumped together in gene phylogenies. Of course, the oxidized sulfur compound used by most modern sulfur reducers is sulfate, and its metabolism came about by very different means. The reduction of sulfate occurs in 3 steps. First, the sulfate is bound to an adenine nucleotide by the sulfate adenyl transferase enzyme, and then it's reduced to sulfite by the adenyl sulfate reductase. Finally, a DSR reduces the sulfite to hydrogen sulfide. The sulfate adenyl transferase is a kinase enzyme, one which is usually involved in the transfer of phosphate groups. More specifically, it's a P-loop kinase, and while most kinases cleave the first phosphodiester bond in an ATP, many of the P-loop kinases cleave the second. One of these is the nicotinamide mononucleotide adenyl transferase, which synthesizes NADH by reacting a nicotinamide with ATP, releasing the diphosphate group bound to the ATP, forming nicotine adenine dinucleotide or NADH. The sulfate adenyl transferase could have evolved from an enzyme which carried out a similar function, and in a manner analogous to the thiosulfate reductase, this would have been facilitated by the similarities between phosphate and sulfate ions. The adenyl sulfate reductase enzyme would have evolved from a flavoprotein oxidoreductase, i.e., one that had a vitamin B2 cofactor. When sulfur respiration evolved, energy metabolism would continue to evolve and advance. All sulfur reducers use cytochromes, the electron transport chain protein with the seroheme group within it, and they use another membrane-bound electron carrier known as the menaquinones, with a redox-sensitive group that is structurally in between that of the menaphenazine used by methanogens and the quinones used by oxygen-respiring organisms. The early origin of sulfur respiration would pave the way for another evolutionary innovation that would define the biosphere as we know it: photosynthesis. While we usually think of photosynthesis as producing oxygen by splitting water, some bacteria carry it out by using hydrogen sulfide as the electron donor, producing sulfate. Interestingly, some photosynthetic sulfur-oxidizing bacteria, or photothiotrophs, use the same enzyme sulfur-respiring organisms use, but they run in reverse. This shared evolutionary history would indicate that one of the pathways was reversed to give rise to the other. If we again look at the gene phylogenies, the base of the DSR family tree is mostly between the reductive ones used by archaea and all the rest. Nestled within the bacterial DSR branch are the oxidative type DSR enzymes used by both bacteria and archaea. This suggests that the first DSR enzymes were used reductively, with sulfur oxidation coming later. Meanwhile, if we look at the adenyl sulfate reductase enzyme phylogeny, the base is between the oxidative lineage 1 and the reductive variety. This suggests that the oxidative and reductive pathways split shortly after sulfate respiration began. The fact that different enzymes are used for the oxidation and reduction of thiosulfate and polysulfide, while the same ones are used to oxidize and reduce sulfate, indicate that sulfur oxidation began as a reversal of the sulfate respiration pathway. But how did this reversal happen? Well, remember that like the functional group in the cytochromes in the electron transport chain, i.e. serohemes, chlorophylls are porphyrin-based molecules. Furthermore, bacterial chlorophylls, which are used by photothiotrophs, are structurally in between serohemes and chlorophylls. Also, while oxygenic phototrophs metabolize water in their light-harvesting protein complexes, photothiotrophs have enzymes elsewhere in their cytoplasm take the electrons from the sulfide and pass them to an electron carrier, which delivers them to their photosystem, where the electrons get energized by the light. As such, one hypothesized scenario for the origin of photosynthesis is that it was originally a respiratory electron transport chain. Energy is a product of this reaction, so if light struck a seroheme in the electron transport chain, it would result in an influx of energy, i.e. the product, which can then reverse the direction of the reaction. A more detailed description would be a photon would strike a metal atom within a porphyrin group, stripping the electron and causing it to go to an electron carrier like NAD, reducing it. Then, the electron transport chain would take an electron from sulfide, oxidizing it. This would essentially be the first photosynthetic reaction, and the seroheme would ultimately become chlorophyll, while the protein it resides in would give rise to all modern photosystems. Now, chlorophylls have a magnesium atom in their center, while serohemes have an iron. So, what did this original photosynthetic pigment possess? One possibility is that it had a zinc atom. This is because electrons can remain in an excited state in zinc for a longer time after being struck by a photon. So, while in an iron atom, an electron that has been photoexcited would often return to its grounded state before being passed to an acceptor at the end of the electron transport chain, a zinc electron could remain excited long enough for an NAD or ferredoxin to to reach the active site of an enzyme at the end of the chain. Later, the zinc would be replaced by a magnesium atom, and the porphyrin would diversify into bacterial chlorophylls and later chlorophylls. As the excited electrons were passed from the sulfide to the NAD, some of their energy would be used by the cytochromes to pump electrons against the concentration gradient. This would then power the ATP synthase enzyme, so light could also be used to power the cellular energy metabolism. As stated before, sulfur respirers use an assortment of enzymes to partially oxidize organic carbon and pass the electrons to sulfate. In order to assimilate carbon dioxide, the NADH would have to take the electrons to a decarboxylating enzyme, reversing the reaction and allowing carbon dioxide to be assimilated as a carboxyl group. This could then be further reduced and transferred to other biomolecules. However, modern photothiotrophs use designated carbon assimilation pathways. Purple sulfur bacteria use the Calvin cycle, also used by oxygenic phototrophs, while green sulfur bacteria use the reverse TCA cycle. The TCA cycle is one of the most ancient metabolic pathways in existence, and methanogens and acetogens both have their own version of the pathway. However, instead of using it to power their energy metabolism or as a means of carbon fixation, they use it to synthesize biomolecules. More specifically, alpha ketoacids, which act as precursors of amino acids along with porphyrins. Within our cells, electron carriers take electrons from various intermediates and shuttle them to the respiratory electron transport chain. This causes it to run oxidatively or clockwise. Two carbon dioxide molecules are released in each turn of the cycle, and these are replenished when an acetyl group on the acetyl-CoA is bound to a 4-carbon oxaloacetate to form the 6-carbon citrate molecule at the start of each turn. However, in the horseshoe TCA cycle, NADH takes electrons from the right half of the cycle, causing it to run oxidatively, and these reduced carriers then shuttle the electrons to the left branch, causing it to run reductively. The oxidizing and reducing branches cycle NAD and NADH between each other. However, once the first form of photosynthesis evolved, NAD and ferredoxins would be photoreduced by hydrogen sulfide, and this influx of reduced carriers would then cause the right branch to run reductively as well. But there is still the issue of the gap between the two branches downstream that has to be bridged if the TCA cycle is to truly become a cycle. This is closed by the succinyl-CoA synthetase enzyme, which produces succinyl-CoA from succinate and coenzyme A. The first division in the gene phylogeny of this enzyme is between thermophilic bacteria and archaea. This would predate the rise of oxygen in the atmosphere, and since anaerobic respirers don't use the oxidative TCA cycle, this indicates that the first cyclic TCA cycle ran reductively. At the end of each turn of the cycle, a citrate is reacted with coenzyme A to form acetyl-CoA and oxaloacetate. The oxaloacetate is used for the next turn of the cycle, while the acetyl-CoA is fed into biosynthesis pathways. Apart from carbon, a major biophilic element that has to be assimilated by primary producers is nitrogen. Modern organisms usually take nitrogen in one of 3 forms: ammonia, nitrate, and diatomic nitrogen. Prior to the Great Oxygenation event, nitrate would have been rare, while ammonia would have been more common, but any organism who took it in this form would be restricted to a source of ammonia. In order to assimilate diatomic nitrogen, the nitrogenase enzyme is required, and this converts diatomic nitrogen into ammonia. Phylogenetic analysis has indicated that this enzyme is indeed ancient, and it had long been believed to be present in Leuca. However, more recent analysis has indicated that it instead evolved shortly after. Whether or not it originated in bacteria or archaea is still being debated, but more publications are leaning toward bacteria, with it being laterally transferred to archaea. Most nitrogenase enzymes possess an iron-sulfur molybdenum cofactor, while some have the molybdenum switched out with a vanadium. In its reduced form, molybdenum is less soluble than in its oxidized state, which means it would have been less common in the oceans prior to the Great Oxygenation event. This has led to speculation that the first nitrogenase enzymes used vanadium, but ancestral state reconstruction has indicated that this is unlikely. Because diatomic nitrogen is held together via a triple bond, it is very hard to break, and the modern nitrogenase enzyme requires 16 ATPs for every diatomic nitrogen molecule metabolized. But did ancient nitrogenase enzymes split nitrogen without the use of ATP, and its inclusion in the reaction just accelerate the rate of nitrogen fixation? Ancestral character state reconstruction has shown that the use of ATP is indeed required for the reaction to proceed at all, implying that the pre-nitrogenase enzyme metabolized a different substrate prior to the inclusion of this step. One possibility is that its progenitor used hydrogen cyanide as a substrate. Indeed, some nitrogenases can convert cyanide into ammonia and methane. To put this all in prehistorical perspective, the oldest isotopic evidence for life dates to 3.8 billion years ago, and the oldest evidence for dissimilatory sulfur metabolism, both respiratory and photosynthetic, dates to 3.5 billion years ago. Apart from photosynthesis, another less prominent form of phototrophy is photoheterotrophy. Here, organisms use light to power their energy metabolism, but obtain material to construct biomass out of from organic carbon in the surrounding environment. Instead of having a porphyrin-based chlorophyll within a photosystem, many photoheterotrophs have a retinol within a bacteriorhodopsin protein. When struck by a photon, it will have a proton knocked off it into the extracellular fluid, and the retinol takes a shape that allows it to take a proton from the cytoplasm, thus generating a cross-membrane pH gradient. It regains its original shape until it gets hit with another photon, and the cycle repeats. Some bacteriorhodopsins pump other ions such as sodium or chloride, and other rhodopsins are involved in other functions, including cell signaling. More specifically, the photochemical reaction with the retinol group is used to detect the presence of light, so the cell can upregulate systems involved in repairing damage caused by UV radiation. Phylogenetic reconstructions have indicated that the original bacteriorhodopsin pumped protons and came about around the time of Luca. This may have been a means of energy generation, but another possibility is that it was used for energy alleviation. When early organisms began to colonize the ocean, they would have to contend with an acidic pH, roughly 6, and thus would have to expend energy to pump protons out of their cytoplasm to maintain homeostasis. Having a bacteriorhodopsin would allow early cells to harvest light energy to pump these ions out, while the energy derived from environmental chemical energy was used elsewhere in the cell. This would be especially true if bacteriorhodopsins evolved before ATP synthase, and cross-membrane gradients weren't used to replenish ATP. Going back to photosynthesis, oxygenic phototrophs have two photosystems, while purple and green sulfur bacteria have one each. Interestingly, the steps of electron transfer in the photosystem II complex in oxygenic phototrophs closely resembles that of the photosystem of purple sulfur bacteria, with the electrons flowing through the proton-pumping cytochrome complex after being struck by light. Meanwhile, the steps of electron transfer in green sulfur bacteria closely resemble that in the photosystem I complex, with the electrons flowing through the cytochromes before entering the photosystem. This implies that the ancestral photosystems diversified into two main varieties before the advent of oxygenic photosynthesis, and that at some point, a phototrophic bacteria with one variety of photosystem obtained the other via horizontal gene transfer. This phototrophic bacteria would ultimately give rise to cyanobacteria, while the two photosystems became the key components in the light reactions in oxygenic photosynthesis. Now, there is still the issue that the purple sulfur bacteria photosystem energizes electrons taken from hydrogen sulfide by other enzymes, while the photosystem II complex splits water directly in the photosystem itself. So how did this switch come about? Well, light doesn't photooxidize water directly, but rather manganese within a manganese-oxygen-calcium cluster known as the oxygen-evolving complex. These are photooxidized from manganese 3 to 4, and the electrons go on to reduce carbon for assimilation. The manganese 4 is reduced to manganese 3 by oxidizing water, producing oxygen, and when another photon strikes the oxygen-evolving complex, the cycle repeats. One possibility is that an intermediate between photothiotrophy and oxygenic photosynthesis was a metal oxidizing phototrophy. The manganese 2 are photooxidized to manganese 3 before being included in the oxygen-evolving complex. While manganese oxidation may have been the immediate precursor to oxygenic photosynthesis, iron oxidation would have been a better candidate for the first form of metal phototrophy. Iron is both less electronegative, i.e. easier to photooxidize, and was more abundant on the arcing earth than manganese. This would have started with an iron 2 that was involved in electron transport in the photosystem being struck by a photon and oxidized to iron 3. The electron would go to an electron carrier to reduce carbon, and the iron itself would be replaced by an iron 2 from the surrounding environment until it was struck by another photon. Later, the original site of the iron would evolve into an active site. In the modern photosystem II complex, the oxygen evolving complex is housed between two subunits, that being D1 and D2. Like the DSR A and D subunits, these two are homologous, meaning at some point they were a single subunit until the gene was duplicated. We can call this ancestral subunit D0, and in the progenitor of the photosystem II complex, it could have taken two ferrous iron ions to produce hematite, or rust. Now, you may have heard that iron oxidizing phototrophs have been identified on the modern Earth, but these are green sulfur bacteria who acquired an iron oxidase gene from chemolithotrophic iron oxidizers, i.e., it's not a descendant of ancient phototrophic iron oxidation. If it utilized a higher energy photon, the pre-photosystem II complex could then oxidize manganese instead, producing manganese 3 oxide. One of the manganese can then be oxidized further to form sodium manganese oxide, producing one extra electron for carbon assimilation. So, now you have a product in a similar redox state to the oxygen evolving complex, which could then later evolve into it. To again put this on a timeline, the oldest solid evidence for dissimilatory metal oxidation dates to 3.2 billion years ago. This process would also provide electron acceptors for the first iron-respiring organisms. The ferric reductase enzyme, which facilitated this, likely evolved from a cytochrome B protein. The flow of electrons in and out of, i.e., the reduction and oxidation, of the iron in the cytochromes would make them predisposed to facilitate dissimilatory iron metabolism. Phylogenetic analysis of the flavin-containing domain indicated that the original ferric reductases were coupled to a hydrogenase enzyme, meaning the first dissimilatory iron reducers used hydrogen as the electron donor and later used organic carbon. Methane, being a product of microbial metabolism, would have been abundant in the atmosphere of the Archean Earth. With the advent of the capacity to reduce oxidized compounds such as sulfate, the oxidation of methane would provide a readily available energy source to any microbes who employed it. Methane is the smallest of the hydrocarbons, and while the oxidation of these compounds can yield a lot of energy because of their reduced state, they are also difficult to metabolize. Carbohydrates and amino acids have various functional groups for the active site of an enzyme to bite into, while hydrocarbons have no such group. The first step of methane oxidation is catalyzed by the methane monooxygenase enzyme, which is part of the multi-component monooxygenase enzyme family. This family is one specialized in oxidizing hydrocarbon-based compounds. These enzymes likely began oxidizing phenols, which would be easier to metabolize because the active site could bind to the hydroxyl group on them. Having evolved a mechanism to oxidize the benzene group, they could then go on to oxidize completely nonpolar aromatic compounds like toluene, or alkenes with their double bonds. Later, some could then go on to metabolize alkanes of shorter and shorter length until they oxidized methane, giving rise to the methane monooxygenase enzyme. The methane is first oxidized to a methanol and then a formaldehyde. At this point, it can be either assimilated or further oxidized to power the organism's energy metabolism. If the latter occurs, it will be bound to the H4-MPT used by methanogens, forming methylene H4-MPT. The methylene H4-MPT will then be oxidized to a formal H4-MPT, and the carbon will be transferred to a methanofuran, again the one used by methanogens, forming formal MF. This is then reacted with water, forming formate, releasing the methanofuran. The formate is then oxidized to carbon dioxide. So, it appears the downstream part of the methane oxidation pathway was acquired from methanogens, specifically methyllotrophic methanogens, who run this part oxidatively. Although, unlike methanogens, methanotrophs use NAD to oxidize methylene H4-MPT and formate, so this part of the pathway can be seen as a mix of methanogenic and respiratory biochemistry. The first evidence of methanotrophy dates to 2.9 billion years ago and used sulfate as the terminal electron acceptor. It was 2.8 billion years ago that we find the oldest definitive evidence of oxygenic photosynthesis. How this came about and how organisms would utilize the oxygen is a topic for another video. In summary, Luca was an early pioneer in harvesting redox chemistry to drive its energy metabolism. Once heterotrophy evolved, it could obtain energy by oxidizing organic carbon. An early sulfur-based respiration was then reversed to give rise to photosynthesis, which used sulfite instead of water as the electron donor to reduce carbon dioxide to produce organic carbon. In modern environmental microbiology, when we look at sediments, we see the redox ladder. Here, the most electronegative electron acceptor is used up in the top layer of the sediment. Below it, the second most electronegative acceptor is utilized until it itself gets used up. As you go deeper into the sediments, the less and less electronegative electron acceptors are used. Now, the oxidized form of the less electronegative acceptors are present in the top layers, but since respiration using the most electronegative acceptor yields more energy, microbes who use the most electronegative acceptor outcompete those who use the less electronegative ones. While using a more electronegative electron acceptor yields more energy per mole of organic carbon oxidized, these compounds also induce more oxidative stress. When organisms evolved ways to cope with this stress, they could then go on to metabolize increasingly electronegative electron acceptors. So, if we look into the distant past, we can see something like a redox ladder, but one that extends through time instead of space, with microorganisms using more and more electronegative acceptors. We can see something similar with photosynthesis, with phototrophs using increasingly electronegative electron donors for primary productivity. The utilization of these more electronegative elements would require cellular machinery which could harvest increasingly more energetic photons. Photons, which like the oxidized compounds, are more inclined to cause cellular damage. But instead of being driven by a greater energy yield, the utilization of more electronegative electron donors in primary productivity would have been driven by variations in abundance. In the Archean oceans, ferrous iron would be more abundant than sulfides, but would be less abundant than water itself. So that's one scenario for how early forms of respiration and photosynthesis evolved. Later, we'll cover how the production and utilization of oxygen came about during the late Archean and the origin of aerobic metabolism. But before then, the next major video, Organic Cosmochemistry, will discuss how the building blocks for life came about. After that, we'll look at how extremophile microbes cope with harsh environments, and then explore the possibility of life on Mars. After we cover the origin of aerobic metabolism, we'll look at how the evolution of various dissimilatory metabolisms impacted and was impacted by the surrounding environment in the ecology of the Archean Earth. Of course, how early life evolved is a controversial topic, with many contradictory bits of information. But trying to piece together what happened back then can be a bit like a puzzle, and if you're interested in it, I encourage you to look through the literature to synthesize your own perspective. That's all for now, and if you like what you see, please like and subscribe since it helps the algorithm out a lot. Thanks.